So here we are, almost there. So this is called How to Interpret Clinical Trial Data. And I'm using clinical trials, not the newest ones, but I think very good examples. So this is really about what you have to keep in mind when you see trial results and have to interpret what it means for you. So what do you have to know? 3 questions: who, why, and how. So why was the study done, who was studied, and how was it done? These are the 3 questions you need. When you look at this, I chose the IMPROVE-IT trial, and not that new trial anymore, but a very good example. So the IMPROVE-IT trial, the question, why was it done? It was done to answer certain questions, and the answers are there, the questions are there. The question number 1 was, does LDL lowering with a non-statin agent reduce cardiac events? So keep that in mind when you interpret it. So that's question number 1. Number 2 is, you know, the LDL hypothesis, the lower the better. The question of this trial was, is even lower even better? So if we reduce LDL cholesterol even further, does it reduce events? And number 3 was safety. You can forget about that, but I would like to keep in mind the 2 first questions. Okay, that's the why of the study. Now the who. Always look for the study population. That's very important. So you can check out the inclusion criteria, exclusion criteria, of course, baseline criteria. So these were patients with a recent ACS. LDL cholesterol was between 50 and 125 milligrams per deciliter, or lower if they were on lipid-lowering therapy. And now here's the how of the study. So how was it done? Okay, what's the study design? That's always important. So you have these patients with a stabilized ACS who had this criteria of LDL cholesterol. 18,000 patients were studied, and those were randomized 1-to-1 to receive either simvastatin 40 milligrams or simvastatin 40 plus ezetimibe 10. Okay, so these were the comparisons that were made. The trial was an event-driven trial, so they were looking for 5,250 events, and it was scheduled, so to say, for 2.5 years. And you may be aware that this trial went on for almost 7 years, so because there was a lower than expected event rate. Okay, primary endpoint you see here, it's an ischemic endpoint: CV death, MI, stroke, that would be MACE. Then you have hospital admission for unstable angina and coronary revascularization. So that's the primary endpoint. Okay, baseline characteristics, always important. Check these out because you want to know who was studied. So typical population, 64 years, a quarter up to a third, normally female population, almost a third diabetic. So that's pretty much representative of the trials that we know. So you can see here what type of event made the patient come in. And then you see the LDL cholesterol, that's important. So the trial started at 95 milligrams per deciliter as a starting LDL cholesterol. Okay, so this is now the effect on lipids, on LDL cholesterol. And you can see here a nice drop in LDL cholesterol in the Simva group and the ezetimibe plus Simva group. And this is that's the median time average. So it's 69 versus 53. So approximately 16, 17 milligrams per deciliter difference. This is also important to interpret the results. Okay, so we were comparing pretty much the 70 goal with something lower, approximately 50. Okay, so this is the primary endpoint in the intention to treat analysis with the endpoint that I just mentioned. And you can see here the Kaplan-Meier curves. There's a significant difference with lower event rate in the ezetimibe group. There's a 6% relative risk reduction, which is significant. This is the hazard ratio you can see here. And over 7 years, the number needed to treat is 50 to prevent one endpoint over 7 years. So my first question, what does this mean in terms of the study hypothesis? So would you say that the trial met its objectives? Remember the questions, the why. So what would you say? Because this is the first thing you have to answer. The trial was designed to answer certain questions. And now my question for you is, did it answer the question?
Certainly confirms the LDL hypothesis.
Okay. So would you say, is even lower even better? The answer is yes, correct. What about the first question? Is ezetimibe able to reduce events? Perfect. Absolutely perfect. So the answer is yes, right? It did answer the questions.
But how lower? Like here it's 16%,
Mm-hmm.
but maybe if it's much lower, then it affects steroids biosynthesis and the hormones.
Yeah. Well, we know from other trials that it does not, but I can only answer, we're looking at this trial, right? So I can tell you, so here we have a certain difference. We have 69 milligrams median in the Simva group, and we have 53 something in the ezetimibe group. So that's what we have in this trial, right? We have very much lower numbers in the PCSK9 inhibitor trials. So, but the point that I would like to make is you have the questions, you have the answers here, and the trial looked exactly for what it wanted, and this is the result. So, we could call it a dream trial. The question now, of course, is what do you do with these results in terms of clinical interpretation? Would you treat a patient? Would you not? Okay, that's a completely different story. Mine is more asking for a formal analysis. So when you look at this, this is a regression analysis, and the plot here shows the level of LDL cholesterol reduction. This is now millimoles, and the reduction of cardiovascular events. And when you look at this, you know, we had 16 milligrams per deciliter reduction in LDL cholesterol. It's very much endpoint reduction, what was expected. But you, of course, as clinicians, have to make a decision if or not you want to use the drug. But in terms of formal interpretation, everything makes sense. Okay, now we have the endpoints. So we have the primary endpoint is a combined endpoint. Okay, then there are some secondary endpoints here that are different combinations of ischemic endpoints. And you see the forest plot here with the point estimate and the confidence intervals. This is the line of unity. You know this type of presentation. My question for you, if we look at the secondary endpoints, is it valid to say these are significant? What would you say? Yes. Why? Okay, so you're saying because they're not crossing the line of unity? That's what you're saying? Okay, I'd say, okay, what if this is not crossing the line, but the primary endpoint is?
Depends on the statistical hierarchy that was set.
Yeah. There's one golden rule. It says the trial is designed for the primary endpoint, isn't it? It's powered for this. Yeah. So let's say if your primary endpoint is not significantly different, the secondary endpoints are hypothesis generating only. But in this case, you have a significant difference. So the endpoint was significantly different, and that's why it's absolutely valid to look for the secondary endpoints. But that's the formal answer to this. Okay. Now we have levels of endpoints. Level 1, all-cause mortality. That's always the purest thing. You count dead bodies. That's basically what you do. Okay, can you improve survival? So you know that we have this in heart failure trials. It's very, very, very difficult to show differences in all-cause mortality in trials with, let's say, ischemic heart disease, for example. But this is obviously the hardest endpoint we have. Then there's cause-specific mortality, for example, fatal myocardial infarction. Then we have non-fatal clinical events, non-fatal MI, non-fatal stroke, hospitalization. Hospitalization is an important endpoint, especially for healthcare and for cost. Then there's surrogates, for example, LDL cholesterol, CRP, something like that. And then it says level 5 is quality of life. I kind of disagree with that because quality of life is a very important endpoint. It's a softish endpoint because you can only check it with the questionnaires and stuff, but it makes a big difference for patients. Think of the heart failure trials where they have sometimes very, very marked improvements of quality of life, and that makes a big difference for the patient, okay? But if you look at this hierarchy, basically all-cause mortality is the hardest endpoint that we have. Okay, so these are the endpoints, the different components of the endpoints here of this trial. You see no difference in all-cause mortality. You have no difference in cardiovascular mortality. There's a lower number of MI rate, a lower number of ischemic strokes, and this is the classical MACE endpoint. There's also a significant difference. Okay. Now, subgroup analysis. You heard about this today? So the subgroup analysis, the rule of the 4 Ps, I like that one. What should it be? Subgroup analysis, subgroups should be pre-specified in the statistical plan. They should be powered well enough to be able to actually have a result here. They should be plausible. It obviously has to make sense to look for certain things, and they need to be practically relevant. So I think that makes sense. Now, when we look at this, major pre-specified subgroups of the IMPROVE trial, male, female, age, diabetic status, prior lipid-lowering therapy, LDL cholesterol high or low. So I think these do meet the criteria. Okay, they're plausible, they're practically relevant. They were pre-specified, and actually they were powered well enough. It's a large trial, 18,000 patients. Okay, so now look at this. And again, you do see differences. We had the crossing of the line of unity. So if you look at this age, it's not crossing here, it's not crossing here, it's not crossing here. Okay, what do you have to look for? Well, it says it there. Okay, don't just look at the lines at the confidence interval crossing. You really have to look for the P where is it here? P for interaction. Okay, and you can see here it says it in this way that there's only a star here for diabetes, non-diabetes. So only for this subgroup there's a significant P for interaction. Okay, whereas there's none for these. So also for this, where you see no crossing of the line, there's no significant P for interaction, which says there is no difference. Okay, but there is a difference, a significant interaction here for diabetes versus non-diabetes. Okay, so that is something important. Now, when we look at this is now a subgroup analysis plotted as Kaplan-Meier curves for the diabetics and the non-diabetics. Okay, so when you look at this is diabetes. This is the diabetic cohort. You see a much higher risk. So you can see this in the Kaplan-Meier curves. You have more endpoints. So there's a difference here. So you can always tell this is a higher risk group very easily. And then you can see there's a 14% relative risk reduction. Keep in mind there was 6% in the overall trial. And there's no difference here for the non-diabetic cohort. Okay, so next question, what does this mean? Only difference in diabetics. Should we only treat diabetics now with this drug? What would you say?
Diabetics benefit more.
Okay. Should we treat non-diabetics?
Overall, the whole population benefits according to
Yes.
the primary.
Exactly. So keep in mind, the study was designed to look for the primary endpoint in the entire study cohort, okay, which include diabetics and non-diabetics. So we cannot take from this, oh, there's no difference here, we should not treat non-diabetics. But it says maybe diabetics do benefit more. But again, this is hypothesis generating. So if we really want to test this, we have to do a trial, a phase 3 trial in just a diabetic population to check if there's more benefit there, okay? That's very, very, very important. But obviously, it gives you some hint that diabetics may potentially benefit more, but it's hypothesis generating. Okay, any questions for this? To one.
It's related that they come in with an absolute higher risk.
Sorry, why is it?
But this is related that they come in with an absolute higher risk.
Yes, but it's not clear from the beginning that they benefit more. This is what we see in many cardiovascular trials. If cardiovascular risk is higher, the absolute benefit seems higher, especially in lipid-lowering therapy. Yes, it's true. But you don't know before you test it, right? Okay, so now we have a change in topic. This is now about atrial fibrillation and anticoagulation. So this is starting with the ROCKET-AF trial, rivaroxaban in patients with atrial fib. So patients were treated with rivaroxaban or warfarin. In this trial, primary endpoint stroke or non-systemic embolism. Okay, this is the statistical methodology. So, yeah, I think we can skip that. This is for primary efficacy, non-inferiority, superiority analysis, and primary safety evaluation. It was bleeding in this case. Okay, you can see the statistical assumptions here. So let's skip that one, and this is the result. So here we have the Kaplan-Meier analysis of the primary endpoint, and you see a difference here between rivaroxaban and warfarin. This is the this is the event rate here. So look at this, non-inferiority clearly met, that's statistically significant. So there's non-inferiority of rivaroxaban compared to warfarin. Okay, Now here's the next analysis. So this is looking in the intention to treat cohort, testing superiority, and in the on treatment. Okay? So you do see it makes a big difference. In the intention to treat analysis, there's no significant difference. So superiority is not met here. Whereas in the on treatment cohort, it is. So what should we do looking at the primary endpoint? Intention to treat or on treatment analysis for superiority analysis? Intention to treat. Yes, so that's the gold standard. It makes a big difference, right? Clinically, for the company, obviously, so it makes a big difference, but yes, it's intention to treat. So we have to keep that in mind. And this is one example why it could be different. So on treatment looks for the treatment period where the drug is given to the patient plus some extra time, which is defined in the protocol. Let's say, I don't know, 1, 2 weeks or 4 weeks or whatever. Okay? So if, let's say, if you have this, if a stroke, and the intention to treat analysis covers the whole study period. Okay? So you know, intention to treat, the patient is randomized, even if he does never take any pill, he's still in the analysis. So let's say if you have the on-treatment analysis and a patient suffers stroke right after that, let's say, blanking period, this would not be counted. Okay? So that's not an event in the on-treatment analysis, whereas it is an event in the intention to treat analysis. And that's exactly what happened in this trial. When patients were switched back from rivaroxaban to warfarin, they had more events. And those were seen in the intention to treat analysis. Okay, but it does make sense to look at it that way. And there's a clear rule. And this says, if you look for non-inferiority, and I think Alexander talked about that this morning, you have the on-treatment analysis, whereas if you look for superiority, you have the intention to treat analysis. Safety is always on treatment because you want to see the bad effects of the drug. Okay? So that's the golden rule. All right. So next trial, the Aristotle trial. So this was same thing, atrial fibrillation patients with additional risk, randomized to receive apixaban or warfarin. Primary outcome, the same, stroke or systemic embolism. Okay, here's again the statistical plan. So if you really want to check out what they did, it's good to read it. You can read it in the protocol, obviously, and you can also read it in the methods and the publication. So primary outcome, first non-inferiority, then superiority, and then bleeding was tested, and then all-cause mortality. So this was the hierarchy of testing. Okay, the results here, primary outcome is stroke events significantly lower in the apixaban group, and safety also less bleeding events. Okay, so I'm just showing that to be complete. And now I'll come back to this. So this trial used apixaban twice daily as the standard dose. This was tested. But in certain patients, 2.5 milligrams were used for certain dose reduction criteria. For example, there was the 2 or 3 rule, and there was kidney function and so on. So I show you this. This is a subgroup analysis comparing 5 milligrams versus 2.5 milligrams dosing in these patients. And you can see here, look at this. This is the full study cohort, and this is the subgroup analysis. 2.5, approximately 5% of the patients had the 2.5 dose. Very large confidence intervals here, okay? But there's no significant difference, no P4 interaction. So my question for you now is, what do we know about this dosing? It is identical, isn't it? So you have 5 milligrams, 2.5, it has the same result. And you may be aware of the fact that in clinical medicine, many patients receive the lower dose, which should be used for dose reduction. So my question for you, is it okay to do that? Or does that mean you have the subgroup analysis here very nicely showing that there is no difference? So is that correct?
Subgroup was too small.
Subgroup is too small, number one. There's one other thing that you have to keep in mind. Who received the 2.5? Was it a parallel randomization? No. Okay, so when you recall the dabigatran trial, they had 2 doses. They had 110 dabigatran and 150, and they had like warfarin, 1 dose, and the second dose of dabigatran, 3 parallel groups. So they tested different doses. Here we only have the 2.5 dose and very selected patients who have dose reduction criteria. So basically, this analysis just means that if you fulfill the dose reduction criteria and use the drug in those patients, they have the same outcome as patients who receive the high dose, which is the standard dose for them. Okay, so if you follow the rules of dose reduction, yes, you have comparable results, but it doesn't mean that if you give a patient who should receive 5 milligrams, you give him 2.5, that he will have the same results. That's a very important thing. And many doctors actually give the small dose because they want to, I don't know, protect their patients to not bleed. But it's not correct to do that. Okay? So all I'm saying here is think of the design. Was it tested in parallel? Were different dosages tested? Or was it, in this case, dose reduction? Because then you have to follow the criteria of dose reduction. Only then results are comparable. Okay, so the last here is the ENGAGE trial. This is testing edoxaban. And in this trial, by the way, there were 3 arms: warfarin, high-dose edoxaban, low-dose edoxaban. So they had like a 1-1-1 randomization. But I'm showing this, and they had dose reduction criteria, which are here. And the reason why I'm showing this this trial is the following. They had something called modified intention to treat. So have you heard about that? Modified intention to treat. So modified intention to treat means the patient is randomized, so he's in the intention to treat analysis, but he should receive at least 1 dose of the study medication. Okay, which means he actually made it into the trial and he received the pill. So after 1 application of study medication, he's in the modified intention to treat analysis. And if you look at this, this is the difference, the numerical difference. So it's a very low difference in this trial. You see like 7,034 and 7,002 actually received the drug, so they are in the modified intention to treat. So it's not much of a difference. You can see it here. Again, my question for you, does it make sense? Should we do this? Basel, this is always a question for you. Why did they do it? Professor Lewis has a microphone, so he can answer with the microphone.
Yeah. It's to deal with the issue of what happens, which I think has happened to some of us, if the patient is randomized into a trial, and it's not a 24,000 patient trial or something like this is, but it's a small trial. We only have a couple of tens of patients. Now the patient's randomized and he doesn't get the treatment. So do you consider him or don't you consider him? And I think it's probably correct to use a modification, which means only get him if he was randomized and took the trial. It can happen. It once happened to us somewhere where a patient with acute MI was about to be randomized or unstable angina. He was scheduled for a cath. He had to get the drug before the cath. While the person was trying to do randomization and get the drug out of the thing, the patient kind of crashed. And everybody said, we can't wait for this. We're going. They took him and they treated, and he never got the drug. Mm-hmm. So it's probably logical not to include him to test whether it was drug or placebo. He never saw the drug. He didn't smell it. Mm-hmm. And they were trying very hard to get a positive result. So they tried every trick they could.
Yes, exactly. But I haven't seen it much after that.
But because the numbers are so small, I think.
Yeah, the difference is quite small.
I mean, how often does that happen? 20 in
yeah, it's not many patients, as you've seen. Just to be complete, I show you, and again, it's the difference of on-treatment analysis and intention to treat analysis. So when you look in the non-inferiority analysis, edoxaban 60 was better, statistically better than warfarin. In the superiority analysis, in the intention to treat, it just didn't work out. It was just non-inferior, not superior. Okay, so again, in the intention to treat, different result than in the on-treatment analysis. Okay, good. Now this is a meta-analysis. Right after ENGAGE, there was this meta-analysis, combining the results of all 4 NOAC trials, which was RELY, ROCKET, Aristotle, ENGAGE, and you can see they always use the high dose for this comparison. This is for stroke systemic embolism. This is for major bleeding, and these are secondary endpoints. Okay, so my question is, what is the value of this analysis? Do we need this? Does it make sense to do it? Is it okay to do it? So what is yes? What do you think? Is there some value of this? Is it okay to do this kind of analysis? The trials were different, the drugs were different. Well, I think it is. What is it?
Okay, but we can check heterogeneity.
Yes,
because,
excellent.
First, if it's like first trial, it's like
Yeah.
the rate or it's more than 30%, but in the second, from my reading, it's 83%
Yeah.
heterogeneity.
Very good, very good. So this is something that you learned from Gianluigi, I guess. So yeah, when you look at this, so heterogeneity is not significantly different in this at this endpoint. It is for bleeding. Do you know why? Why there's heterogeneity? It's a very simple answer to this. Exactly, because bleeding definitions were so different. Okay, but what does this meta-analysis do? Why is it? I think it has a value. To compare NOACs versus vitamin K antagonists. Exactly. It's looking for a principle, right? It's comparing a new principle with an old principle, if you wish. And I think that makes sense. And a good reason to do a meta-analysis is always to look for rare effects, okay, for outcomes that have lower numbers. For example, intracranial hemorrhage, that's a very excellent example. It's something that doesn't occur very often. But obviously you have more events in a meta-analysis, and then you may have different effects in terms of significance. Another one is mortality. Okay, to have a significant effect on total mortality, you need to look very long. You need to look in many patients, or you need to have very, very sick patients who die more frequently. Then you will have an effect. So meta-analyses use the higher sample size to also look for effects on mortality. If you do this, you can check it out here. This is all-cause mortality. This is actually significantly different in this meta-analysis, but it was only in one of the original trials. Okay? So I think it does make sense to use it. Okay, so here I have something different. It's the COMPASS trial. It's also using a NOAC, but in ischemic heart disease, in PAD and in coronary disease. And this was testing the value of add-on rivaroxaban 2.5 BID to aspirin, so a very low dose. Okay? So the groups were rivaroxaban 2.5 plus aspirin, rivaroxaban 5 monotherapy or aspirin monotherapy. Large trial, 27,000 patients. Okay, this is the outcome, the primary outcome. So the endpoint was MACE, cardiovascular death, stroke, MI. And look at this, hazard ratio 7.6, significant p-value. So first question, sorry, I have to go back, I was too quick. So this is the primary endpoint. And now you look for the components, cardiovascular death, stroke, MI, and look at the p-values here. So my question for you is, it clearly met its objective in reducing the primary endpoint. That's absolutely clear. So now my question for you is cardiovascular death significantly lower? Yes or no?
Yes, because of primary endpoint.
Right. Perfect. Exactly. That's it. Okay. So you can go to the components, and it's exactly what it is. So death was reduced, cardiovascular death. Okay. Now here's something else. Here are secondary endpoints, and you have to be careful here. This is looking for ischemic endpoints, and I have all-cause mortality here. So it is lower, 0.01. So what does that mean? Looks good, eh? Be careful. When you read this paper, you have to really go into the statistics. It was quite nicely covered, if I may say so, in the paper. But they had a definition, the threshold p-value, okay, for the above comparisons was 0.0025. Okay, so here's my question. Is total mortality significantly different? No, it's not, but you really have to be careful. They hit it quite nicely in the paper. Okay, but from a statistical point of view, yes, mortality is reduced numerically, but it's not statistically significant. Okay, because of that definition. This was the definition. They said hierarchical testing, okay, and they said only if we meet this p-value, then we consider it statistically significant. Yeah. Well, it might not be chance, we don't know, right? We don't know, but it's from a formal perspective, there's no difference. Okay, so watch out. Good. And now here's the OSLER program. So the OSLER program was testing evolocumab in very different patient populations in phase 2 and phase 3 trials. Okay, so there were patients who came out of the OSLER trials. That's the OSLER program. So patients were tested in different studies using evolocumab in phase 2 and phase 3. So patients coming out of that were asked to then be enrolled in the OSLER study. So I have to say it that way. Okay, so you can see they have different names here. So patients who came out of this were asked, would you be interested in another study, which was then called the OSLER trials. Okay, so 4,465 patients were elected to be enrolled in a, let's say, follow-up study. So there was a washout period, and patients were randomized 2-to-1 to receive evolocumab or standard of care. Okay, and the data of these two trials, we have to say, were combined in one data analysis. So what do you think about this design? Is it okay to do that? I mean, can they do it like this? What do you need to know?
What's the standard of care?
Yeah, the standard of care is like, it was placebo plus whatever everybody gets. But they took patients from trials in which patients were enrolled to receive evolocumab. So they took out the evolocumab-treated patients who actually tolerated the drug, okay? And those patients were then, after washout, randomized to receive placebo or evolocumab. It's quite a smart idea, isn't it? Because you have patients who you know, number one, stay in the trial, and number two, tolerate the drug. It's so quite smart. My question for you is it okay to do that? Or do you need to know something else to answer this question? You should ask me, what is it looking for? What is the trial design? What is the endpoint? Because then I can give you the answer if it's okay or if it's not okay. Number one, I think it's a very, very smart idea to do it like this, because you select your patients in a positive way, that these patients actually tolerate the drug, stay on the trial. So you have to know what OSLER was actually looking for, what was the endpoint. And the important thing, it was a safety study, and that's why it is okay. Yeah? So this is what OSLER looked for. So the primary endpoint was safety. Secondary endpoint, LDL levels, and then there was a pre-specified exploratory outcome, which was cardiovascular events. Okay, so it was not looking for outcome, it was looking for safety, and that's why it is okay. Okay, so just to be brief, this is the safety. Safety was good. There was some a little higher neurocognitive adverse event rate in this study. LDL cholesterol was nicely reduced, 60% relative LDL cholesterol reduction. And now here's the cardiovascular outcomes. You see the Kaplan-Meier analysis here, and you see a hazard ratio of 0.47, saying evolocumab reduces cardiovascular events. Okay, so what does this mean? You know, it's great, isn't it? It's 50% endpoint reduction. So does it mean we should prescribe this drug to have endpoint reduction at this stage?
This means that there should be a large phase 3 outcome trial?
Right. This is a pre-specified but exploratory outcome. Okay, so it wasn't powered for this. The trial was not designed for this. But obviously it's quite convincing to see this. From a company perspective, I guess you're quite happy if you have this. And obviously, yes, you're right. You have to go into a phase 3 trial looking specifically for cardiovascular outcome, powered that way. Absolutely true. And this is what happened. This is what you know. It's the FOURIER trial, 27,000 patients, evolocumab versus placebo. These are the results. Primary endpoint significantly lower in the evolocumab group. But look at this. There's a 15% relative risk reduction, not a 50% relative risk reduction. Okay, so there's quite a big difference. But it's exactly that. You have hypothesis generating. You have to go for the phase 3 trial. Okay, excellent. So here's the ODYSSEY outcomes, which tested alirocumab versus placebo in 19,000 patients post-ACS. I just go quickly through this. This is the primary endpoint, different definition of MACE. And again, you have a 15% relative risk reduction here for this ischemic endpoint. This is the phase 3 trial for alirocumab use. Okay, and this is now the secondary efficacy endpoints. And keep in mind, there's hierarchical testing. That's why it's set like here. So you always check the primary endpoint first, and then in a hierarchy, you go down in your statistical testing. Okay? So what I would like to show you here is, for example, this is the MACE endpoint, which is reduced. This is cardiovascular death, not reduced, and now here's all-cause death, 0.026. So is this significantly lower? I can't ask you. So again, the question is, what does it mean really? It looks quite good, but look at this. It's a nominal p-value. It's hierarchical testing. It's very much down bottom. It wasn't done for this. And I don't think you can take from this trial that alirocumab reduces all-cause mortality. Number two, it's not really plausible, I have to say, because cardiovascular death was not reduced. But just be careful, because it's very much at the bottom of the hierarchical testing, and that's why it says nominal p-value. And there's one other rule. If you do your hierarchical testing and the endpoint that is not statistically significant kind of stops your analysis, right? You shouldn't go further because there's no significance anymore. They did show the p-values though. Okay? All right. All right, good. And here's another one. I love this analysis, I have to say. This is a subgroup analysis looking for different levels of LDL cholesterol at baseline. Okay? So you see here the different Kaplan-Meier curves below 80 milligrams per deciliter, 80 to 100, larger than 100. Difference, more benefit here in this group. You can see it here in the forest plot. And you can see the p-value here. So, what do you think about this subgroup analysis?
It's valid.
It's valid? Is it significant?
Well, it's significant. The primary endpoint is met, and it's very plausible that the higher the risk, the more benefit.
What do you think of that? P for interaction 0.09? It's not significant. I totally agree with you, though, and I want to have Basil's opinion on that. But again, from a formal perspective, it means there's no difference, which means you can in all of these subgroups, you can assume the same effect as in the total population. This is basically what it means. But I have to admit that I agree with you. It's very plausible that patients with higher LDL cholesterol benefit more. But from a formal perspective, there's no statistically significant difference. And I would like to hear your opinion on that, Basil.
I absolutely agree. You could say hypothesis generating, logical, but not proven.
Although it is very plausible to think, also from a biological point of view and from everything we know from cholesterol, that patients with higher LDL cholesterol have higher risk and therefore may benefit more. I completely agree with you. But from a formal perspective, there's no statistically significant difference. Okay?
So, okay, but you could do the other way around, like we did with the diabetes.
Yeah.
Because if you said that was significance, then maybe only for those and not for others.
I agree with you.
What you learn from this is that it's good for everybody, irrespective.
Yeah. But again, if you want to know hypothesis generating, then you have to do a trial in patients with LDL cholesterol higher than 100, and then you know, which will not happen. Okay, good. So we have this. Now I have the last thing, which is completely different. This is now about devices. So this is about PFO, persistent foraminovaly, open foraminovaly, patent of foraminovaly, closure in patient with cryptogenic stroke. This was the RESPECT trial, one of the first trials that we have. So patients needed to have a so-called cryptogenic stroke, which means the neurologist did not find a cause for stroke. And they also needed to have a PFO and a transesophageal echocardiography. Okay? And these patients who did not have other reasons for stroke, so no carotid disease, no atrial fibrillations, were enrolled in the trial to test whether closure of a PFO with a device is superior to medical management in terms of prevention of recurrent stroke. Okay, this is what was tested. So what was done, so this is the occluder device that was put in, approximately 500 patients per group, which was actually the largest that we have on this topic. This is the medical management group, and there was a bubble study, a TE study at 6 months. Okay, so this is the results: safety, very safe procedure, no increase in atrial fibrillation, no difference in bleeding. There were more vascular complications. For those of you who do not know the procedure, it's a percutaneous procedure where you have to go through the femoral vein. So you have to do a puncture in the vein, and you have to move your device up to the heart. Okay, and obviously this produces problems in the group that received the treatment, and obviously not in the group that did not receive this treatment. Okay, so technical success means the device was put in, it worked, very high. Procedural success meant there was no safety issue within a certain period of time, and then effective closure was tested in a TE study after 6 months. So you can see it's a very effective procedure. Okay, now here's the intention to treat analysis of this trial. You can see here the device group up here and the medical group down here, and you see quite of a difference here. Relative risk reduction 50%, 50. You see the very small numbers, keep that in mind. So the event rate very low. Okay, it's a rare event to have a second stroke. And then you see the p-value 0.08. So what is your interpretation?
What's the slope rank?
It's just the statistical method. But it's just look at the p-value 0.08. So that's not significant. So it means, exactly. So it means device closure is not better than medical management, right? So that's quite something. Okay, good. Now here's the sad story about this trial. It's very rare to have a second event. But in this trial, people on the waiting list, they were randomized and they waited to receive the procedure. 3 out of 9 of these patients had a stroke while waiting for the device. It's absolutely super rare, but it happened. Okay? So these 3 patients were counted as a stroke, but they didn't have a device put in. Okay? But it's intention to treat, right? It's randomized. So you're randomized, you're analyzed according to the group that you were randomized to. But these patients never had a device in. Okay, when you look at the as-treated cohort, which means patients actually received a device, you still see rare events, but you have a highly significant result here, 73% reduction. Okay, so that makes quite of a difference. And this is something they call totality of evidence. When you look at this, this is intention to treat, not statistically significant, per protocol as treated. So in the as treated cohort, very much of a difference. 5-year number needed to treat 23. So that's everything you would want. But the formal analysis means there's no statistically significant difference.
I guess this is where you wanted a modified intention to treat definition up front.
Don't say it now.
Modified would mean that you would only randomized when you were randomized and received the first dose.
Yeah.
In other words, the device.
Yeah. I will ask this question again. But yes. So now here's the end of the story. You might be aware that actually there was a longer follow-up of these patients, and new trials were designed where they learned from this trial. And we now know, we have 3 different trials, that PFO closure is actually effective in terms of recurrent stroke prevention in certain patients with PFO. Okay, so we do know also from this trial and a longer follow-up that there was also in the intention to treat analysis a significant reduction of strokes. Okay, so here are 2 questions now. So when you look at this is a meta-analysis of the randomized trials. This is with the so-called Endpoints occluder. This is the Starflex occluder. And when you look at this is the Endpoints device, and this is the Starflex, which is like a spidery, nasty device. I actually put it in when I was doing this. And here's my question now. Here's the test. So this is the meta-analysis, and here's the test for heterogeneity. And when you look at this, the p-value is not significant. So there is no heterogeneity between these groups. Now my question for you is, how can that be possible? Look at these nasty looking devices. They're completely different. So how can there not be heterogeneity? What is the answer to this?
The same effect, same mechanism,
Yeah.
same subgroup, same design. That's
Yeah.
why this is also a different
Yeah.
different form, but have same mechanism, same effect.
Yeah.
That's why it's not
Absolutely. Again, it's comparing principles. It's comparing the principle of closure device versus medical management. And that's why you're absolutely right. That's why they look different, but also NOACs are different molecules, right? But it's looking for principles. And this is why there's no heterogeneity. Good. And now here's the next. So when you look at this, this is stroke TIA in the intention to treat analysis. These were the first 3 trials looking for this. Now we have 3 more, or 1 follow-up analysis and 2 more. But when you look at this, this is the significance level not met. You see the confidence intervals here. So when you look in the intention to treat analysis of these 3 trials, there's no significant difference. When you look at the as treated analysis, so again, the patients who actually received that device, there's significance here, okay? So, and this is exactly what Basel just said. So this is just a question, it's more a comment. If we should really use intention to treat analysis in this type of trials, it is the gold standard, and it is done that way. And if you have, let's say, trials with more patients, let's say in stent trials, and maybe it makes sense. But like we said, if you have the device not put in, what kind of analysis is that? You cannot compare it. So that would be the question is, should we use intention to treat or as treated? And then there's the third possibility, and I think that would be a very fair approach, modified intention to treat if you wish. Because if you do an interventional procedure, you attempt to do something and you may harm a patient. If you poke his groin, you go up to the heart and you do some damage and you never put that device in, but you still did harm to the patient. So that should be also be in an analysis. And that could be kind of modified intention to treat. So the patient actually makes it to the procedure, you start the procedure, you don't succeed, but that patient would be in. So this is more or less just a comment, but please keep in mind that the gold standard also in device trials is intention to treat analysis. Okay, good. So we are done with this part, and keep that in mind. Join our working group. Here it is, that's this. You would just need your CV, make sure you're in the medical field and no robots, and please join our group. Any questions from your side?
Yes. In the paradigm trial, which is obviously an outcome trial, there was a run-in phase where the 4 weeks received ACE inhibitors, 4 weeks received therapy, washout, and then they were randomized. Is this correct to do this?
Well, it is. Of course, it is correct. It's just it limits the results in a way that you have a more selected patient population. But of course, it's correct to do it. I mean, they specified it. The reason to do this is obviously to have more patient in the trial without side effects. And this is what they did. They looked for side effects in the run-in phase. Okay, so they wanted to exclude patients. Well, they excluded patients with previous side effects to these drugs, well, to ACE inhibitors, right? So, but then they did this run-in phase for safety reasons and to see whether or not the patient tolerates the drug. And then they tested both drugs, and then they were randomized. So this is pretty much ensuring that you have a higher number of patients who actually make it through the trial. But again, the interpretation is exactly what you do. So if you think of it in interpretation of clinical trials, this is a limitation, and that made it into the guidelines. So the guidelines said only a patient who tolerates ACE inhibitors may be switched. This is actually what the guidelines say, because the study design was like this. But of course, it's correct. You have to specify it. And then you have to keep that in mind when you interpret the trial. Okay, anything else? Yes?
In an equivalence study where both treatments, I want to say they are the same, which analysis would you prefer, intention to treat or superiority? On total?
If you just want to check if they are the same, it's non-inferiority analysis. But if you want to, like you said, if you want to check for superiority, is one drug better than the other, then you have to use intention to treat analysis. If you want to check for, well, non-inferiority, it's the word, you just want to see if treatments are the same, then it's non-inferiority analysis,
So,
which is in the on-treatment cohort, not in the intention to treat cohort. Okay? All right, are you ready for the last bit? Okay, so we need the last presentations, gentlemen. This will not be very, very long. Okay, so these are just some thoughts about how to write a manuscript. I'm not sure who of you actually writes manuscripts? Oh, there's some. Okay, very good. So that's maybe why you're interested in that. Okay, so look at this, the 7 deadly sins, publish and perish. You know the words publish or perish, which is a common principle at university. But there's publish and perish as well. So if you manipulate your data, you invent your data, you duplicate, you steal, you have redundant publication. Obviously, if you do this, then you will perish because this will not be forgiven. Okay? Then conflicts of interest are something that always have to be declared. And obviously, if there are concerns of the human use in your study or the animal use, the study is useless in a way, you will not publish that. Okay, but I think this is one part that's very important because there's much pressure in the systems to publish, but you should not be tempted to manipulate your data. And you know that there's always cases of people who actually completely invent what they publish, which is quite amazing, but it is possible. Okay, so statistics, use statistics to make most out of your data. What is the most appropriate to use it? Okay, and if you have a statistician or you have an institute at your university, for example, use their knowledge, okay, and ask them what is the best method to actually get the most out of my data, and that's okay. But you should not use statistics to manipulate your data, and that's this nice thing. There are lies, damned lies, and statistics, and as you know, you can do everything with statistics, but you should use the appropriate statistics to make the most out of your data, but not manipulate. Okay, what is redundant publication? Same data, different journal, that's quite easy. Data on website, very difficult. So let's say if you put some of your data on your website because you want to show how great your working group is, difficult, be careful with this. Conference abstract is fine. So if you send your abstract to a conference, that's always no problem. That's no redundant publication. Data and review article, very difficult. If you're planning to publish original data within a review, you have to ask before you send the original data out if that is okay or not. Otherwise, you might have a problem because it's viewed as redundant publication. Good. What is a good paper? Good science, obviously. Innovative, that's the most important thing. You have to have a good idea, solid data, and this is something quite important. Good writing. You have to be good in your manuscript because you always have to keep in mind the guys who review your manuscript don't take much time. They get many, many, many reviews that they have to do, and if you look at a manuscript that looks terrible, just the language and the style and the appropriateness of, let's say, figures or stuff, you don't think that it's a good paper because you think maybe the science behind it is the same quality. Okay, so you have to have good writing and good style and everything. Okay, so what could that be? Innovative therapies, mechanism of action. I think that's quite clear. Not all of us are so lucky to have a very innovative new topic. But obviously, if you have something like this, unmet medical need, and also in science and basic science, whatever, that would be perfect. What is a good journal? There are measures, immediacy index, and obviously the impact factor. This is important, but a little bit unfair. If you work in a field, let's say ophthalmology, the impact factor is never high because it's not cited as much. It's not really fair. So you have to compare within this group of medical field what has a good impact factor. Okay, obviously, if you publish in the England Journal of Medicine, it has a very high impact factor. You can be sure that is a good journal. Okay, So those who have the most to say it with the fewest words. Okay, obviously you have to write things into your manuscript, but you should not be overwhelmingly long. Manuscripts do not get better with this. And if you've ever written a manuscript that is long and the editor said it's fine, but you have to shorten by half, you think it's not possible. But in the end, you do succeed and you have a very good manuscript, you know, so it is true. So think of what you say and don't make it overly long. Good, so what is this? Organize the results in a logical sequence, so it makes sense the sequence of your figures makes sense. Really think which data you want in figures. You have a limited number of figures. Obviously, you can put them in online supplements and stuff, but within the manuscript, you should think first, what do I really want in the figures? And I think this is important. Number one, know the focus of your paper. What do you actually want to show? What is the main thing? And you should discuss, okay, what is really important? Because you might be completely stuck in your own little world of this, but somebody else might think a different way. So if you show it to somebody, to friends and colleagues, some people who are close to the research field and also people who are not that close, they might give you good hints about what is really the key point and what doesn't make sense. If somebody reads the manuscript and he doesn't understand what you're saying, it's never good. Because if they don't understand, other people, the reviewers, might not understand as well. Okay, so what are the most important findings? They are the central theme. Very importantly, if you think of a journal, you have to think about the readers of the journal. What is the scope of the journal? Who's reading this? And it should be written according to, number one, the style of the manuscript, the journal, but also for the readership of this journal. Okay, and again, you see it here, prepare everything carefully. That also shows the quality of your work. Okay, the title. I remember I had this one thing. We had a manuscript with a short title, and we sent it in, and we got a review back and say, hey, this is a good paper, but we want this title. And the title was completely long. It was terrible, but we did it, obviously, because it was accepted. Normally, it's the other way around. You should have a short, catchy title. And it's not always possible, but I think that's important. And I think the New England Journal of Medicine is a wonderful example of how it can be done. They have the most striking titles, you know, just in a few words, everything is said. So that's always good. Okay. Authors. Include only authors who have contributed, which is sometimes difficult. You know, you may have a very long author list because you have friends and they need to be on, and, you know, your chief and whoever. And you know that journals nowadays ask for the contribution of authors, so you have to specify who actually did what. So basically, you have to think of that, and you should only include authors who actually worked for this. It doesn't mean that they have to be to do the data collection or the animal experiment or whatever, but they have to have done something, at least read the manuscript and give you good advice. Okay? But so include only authors that have contributed to the journal. Okay, this is the structure. I guess you know all this. This is kind of always the same pretty much. So you know this, and I think this point is becoming more and more popular. If you work in basic research, reviewers ask for many additional experiments, and you cannot put them in your paper, and you always put them in online supplements. And you know this from big trials. So many things come out. The New England Journal has, 3 figures, and they have supplements with, I don't know how many supplemental things. So I think it's more and more common to have additional online material. But the style, the manuscript structure, I think has not changed much. It's always the same. So materials and methods, you have to write appropriately enough, okay? So too little is always bad because the reviewer will say, hey, what is that? I don't understand that methodology. But on the other hand, you don't have to explain everything new if it's a standard procedure, okay? Unclear flow of procedures is never good because people who really want to know what you did, they read it and say, oh, I don't understand that. What the hell are they doing? It's never good. People should understand what you did, okay? Good. And this is something about style in general. Too many, too complex sentences. You know, some people like to write that way, but Thomas Mann style of writing is maybe not what you should do. It's, you know, scientific language is rather short. Okay, good. So materials and methods should not have results in it. It's quite clear. Results. Objective presentation is kind of the summary of the data. You should not discuss at that point. You basically just, in a logical sequence, show your results. Redundancy doesn't make sense. You don't have to write everything in a paragraph, then have it as a table, and then have it as a figure. That doesn't make sense. You should not discuss at that point. There should be no figures within this. It should be in the figure section. And no methods, materials, they belong to the method section, obviously. So you just report what you find, and you reference your tables and your figures. Okay, now the discussion. First point, you don't have to do it like this, but many do this. You have like the first sentences just summarize the main findings of a study. I find this nice because you go into this and they said, this study shows la 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 la. I think it's nice. You don't have to do it that way, but I think many people do it. But then you have to interpret it. That's the most important thing. You know, what is your hypothesis and did it actually confirm it or not? You know, is there any alternative hypothesis? Because some good research actually doesn't show what you wanted to find, but you have something completely new, but that's great sometimes, but you have to actually analyze it and discuss that, okay? What interpretation can be made? Is it in agreement with previous research? What is a potential reason why you did not find this, and is there any source of error? You know, so you have to kind of mention that. And what does it mean for the field? Very importantly, do not draw conclusions that are not substantiated in the results. That's the worst thing you can do. The reviewer reads it and say, that's very speculative, but you know, they cannot prove this. And then you say, this is a nice paper, however, the conclusions cannot be made, bye-bye. It's not good, okay? Common mistakes. Mix it with results. Okay, you don't have to do that. Discuss results that have not been presented. That's also something, okay? You can only discuss what you have actually shown. Too broad statements, and I have it here, conclusions not substantiated. That is a problem because it leads to rejection of a paper. If you state something that cannot be concluded from your data. Incorrectly discussing inconclusive results is a problem. Inconclusive results are not a problem. This is science. You know, you never know what you get. Sometimes it's very easy and sometimes it's not, but then you should discuss it the way it is. People will appreciate what you write and your results may be important, but you should not incorrectly do this. Good, missing information. Okay, that's something we don't want as well. Conclusion part. That is a short summary, of course, very short. And this is now style. Do not write the same conclusions of the abstract. You can do it whatever way you want, okay? But normally the style is a little bit different. Okay, acknowledgements. Remember to thank the funding source. Very, very, very important. If you ask for funding, let's say for pharma funding, for an institution, for university, whatever, you have to name these people in there. Otherwise, you will not get funding anymore, okay? So in your manuscript, the funding source needs to be there. Okay, acknowledgements. So if you have people who really contributed to the paper but do not qualify as authors, you should mention them. And classically, this is technicians who do a lot of work, study coordinators, and also colleagues who do not really qualify but gave you maybe very good advice, but you think they're not qualified as an author. But they read your paper and said, okay, you know what, you should think about it that way. So this is something that should be acknowledged. And this is a very prominent example. Maybe you know these guys. This is the Nobel Prize for DNA structure. It's Watson and Crick, these gentlemen here. And they actually acknowledged other people who also were on the same track, but they were faster and they got the Nobel Prize. So they were the lucky ones. Okay, so before you submit, and that is something very important. I had a very pushy chief, and I remember that he always said, I submit. I think you should not do it. Leave it there, let it rest. You need some distance to that, and then you should read it again yourself. And then go through, and then sometimes you will find something that you wrote and say, oh, that doesn't make sense. I don't understand it myself, so you can clear up that manuscript. Get feedback. That's what I said. Some people who are close to this and a little bit further away from the topic, so they may give you advice. Okay, somebody should read the whole paper. That is important because, like I said, if somebody reads it and you say, I don't understand that, it's not good. And it should be read also in terms of style and stuff like this that you may not see because you're blind in a way after weeks and weeks on this topic. And then here's that thing, and it sounds so clear, but really check for your spelling, your grammar, consistency in abbreviations. Are your tables accurate? Are your figures accurate? Is your paper accurate? So don't be sloppy. I cannot stress this enough because you may have a great paper. If it looks crap, okay, in terms of style and in terms of accuracy, reviewers may just reject it for that because they think, what is this, you know? And you think instinctively that the quality of the manuscript reflects the quality of your research, and you may get rejected for that. So really take your time to go through this and have a perfect paper when you submit it. Okay, now your paper gets in revision. You get it back. It's not rejected, and even if it is rejected, you get it back. Read everything. Okay, read everything they write. Some reviewers may appear as idiots, as ignorant people who do not understand the brilliant science that you have done. It might be true. Maybe they're ignorant idiots, okay, who do not know what they wrote and they had problems with their wife and that's why they rejected. You don't know. But mostly, reviewers do spend some thoughts on this and they may have some good points that can give you. So, number one, read it carefully and think about it if it makes sense what they write, yes or no. If you think the reviewer is an ignorant idiot, don't write it that way, please. You should make it sometimes you have to make it more clear what you mean, and then the reviewer will understand. Be polite and respectful if you disagree with the reviewer's comment, but don't be submissive. If you think you are right and he is not, you can make your statement. Be always polite. Don't say, you ignorant idiot, you don't understand what I mean. But you can write it. If you have the feeling that it's not correct, make your point. That's fine. And of course, it's always a point answer that you should give. And sometimes it's good to have additional results who you just give to the reviewer. So you can do that. If a reviewer asks for something, you have an additional graph, put it into the reviewer comment, but not into the paper. That's something possible to convince the reviewer. Okay. So why is it rejected? Number one, maybe the journal is not the right one. Okay, you send it to a journal that has a different scope. You thought it's good, but maybe it's not that. Second point, make your paper more sexy. Maybe you have to present your results nicer. Okay, so actually people understand more why your paper is so great. Obviously, you have to critically revise it. So think about the review comments. Does it make sense? Can I improve the manuscript? Sometimes more experiments, for example, if you do a basic research thing. And you can also use preliminary stuff. Let's say if you have a manuscript that's rejected, you can sometimes use it for grant application. So it's worth something. Okay, and here's the process. So you complete your research, you prepare your manuscript, you submit, you have a decision, then you have a rejection. Rejection is not always rejection. Sometimes it's absolutely clear that you have no chance. But sometimes you do have a chance. Maybe you put in additional data, you rewrite it, and you can still write to the editor and say, look, we've gone through this, and we do think it is appropriate because of that and that and that. Please give us a new chance and re-evaluate our manuscript. And sometimes it helps. You can try. There's not more that can happen that as a no. I say, no, we will not do it. But sometimes it helps. Okay, if the decision is accept as is, that's very rare, I have to say, that's great. But if you get a revision, you have to revise it, resubmission, re-review. You still may reject it, and it still doesn't mean anything. You can still go back. At some stage, you should really think, does it make sense to go on? I remember having a basic science paper that I think we spent a year on additional experiments. We answered every single question, and then after a year, they got a third reviewer, and this guy just killed the paper, and that's it. That was a 1 year in vain. It wasn't in vain. It went to another journal, but things like this may happen, and maybe we shouldn't have done it. Okay, if it's accepted, that's great. So, if it gets rejected and you don't think you can improve it, you should send it to a different journal. And think about the journal you should send it to. And I have a suggestion for you. If you have great research, you can send it to our journal, okay? Which will get an impact factor. Basel said it on day 1. So this journal will get an impact factor, I think, this June, if I'm not mistaken, which will be high. We think about 5, which is absolutely great. So if you have great science, it has to do something with cardiovascular pharmacotherapy, you can send it to our journal.